reminder, there's a test on Friday, which is the announcement, and that it covers everything that's covered up until Wednesday. So the 4 lectures, 2 lectures last week, 2 lectures this week are going to be on the test on Friday. So let's get through some of that stuff. So we were talking about striated muscle contraction, and there's a structure called a sarcomere. It's a repeated structure, and it's largely made up of actin microfilaments and myosin 2 thick filaments, and we talked about how myosin 2 walks towards the plus end of the actin, right? So this structure, that's a sarcomere, is a stable structure. So in striated muscle, you do not want your actin depolymerizing. So there's a lot of other proteins that are present that regulate things within the sarcomere. So it's a very complex structure, and this is sort of revealing some of that complexity, right? So you have these structures called Z discs that are sort of central to where the actin is polymerizing from these Z discs. The actin is then sort of these actin fingers are coming out from the Z disc towards the middle of the sarcomere, and the actin is capped at both sides. So the actin is capped by tropomodulin on the minus side and cap Z on the plus side, so the actin is stable. Within these actin fingers, then, you have myosin thick filaments, so myosin 2 thick filaments that are centered in between these actin fingers. The myosin, because it's a plus end directed motor, it will try to move to the plus end, so it's going to try to move towards that Z disc on both sides during contraction. So during contraction, the Z discs come closer together, the I bands shrink, but the A bands do not change size because the A bands are the myosin thick filaments. Other proteins are involved here that are involved in stability. So there's a protein called a nebulin. Not only is actin capped at the plus and minus end, but it's also coated by a bunch of other proteins. One of them is called nebulin. Nebulin also there to help stabilize that actin. The myosin thick filaments that's sort of in the middle of the sarcomere aren't just floating there. They are held in place by a number of proteins, and an important protein there is called chitin. So there's this protein called chitin that's holding the myosin thick filament in the middle of the sarcomere. This chitin, because it's holding on the myosin thick filament, it's holding myosin thick filament in place. As the myosin thick filament moves during contraction, the myosin thick filament is moving towards the Z disc. Chitin has to be compressible. So chitin not only holds the thick filament in place, but when the myosin thick filament is moving, the chitin, which is sort of in the I band, is going to be compressed. So that's what these little squiggly lines are trying to represent, is that chitin is a protein that's not only holding the thick filament in place, but it's also going to get compressed during contraction. Okay, so during contraction, chitin is compressed. During relaxation, it doesn't really push things apart. So how do you separate the Z discs after the contraction? There is a little bit, once the myosin lets go, the chitin does kind of relax and push things apart, but most of the muscle relaxation is actually muscle on the other side opening that up, opening those sarcomeres up. So if you flex with your biceps, you're going to open things up with your triceps, for example. So usually there's opposing muscles that help to open up sarcomeres once contraction is closed. Okay, so you have actin and myosin interacting. We know how myosin uses ATP to move towards the plus end. How is this regulated? Muscle contraction, striated muscle contraction is voluntary. You choose to contract those muscles. Okay, so striated muscles are voluntary, and they work through nerves, and these nerve impulses trigger this contraction. Okay, so your muscle cells are innervated. So in your muscle cell, you have repeated structures, these sarcomeres, right? So you have the Z discs and repeated structures, sarcomeres that are found in your muscle cells. When you want to contract, you're going to send a nerve signal, and that nerve signal is going to be responsible for releasing calcium. How does it do that? It does that with the help of these structures called T tubules or transverse tubules. Okay, so muscle cells are large cells. They have a plasma membrane. It's called the sarcolemma in muscle cells, but basically the plasma membrane. There are extensions of the plasma membrane that go deep into the muscle cell. So these structures called T-tubules that basically are part of the plasma membrane that invaginate in. They get pushed in, or they are sort of structures that look like they're pushed in. So the plasma membrane has these transverse tubules, T-tubules, that are deep in the muscle cell, and they come very close to the sarcoplasmic reticulum. The sarcoplasmic reticulum is a modified endoplasmic reticulum that's found in muscle cells, and in it is stored calcium. So this is sort of a more realistic representation of how things look. However, if you want to look at it simply, this is sort of what we have. You have a muscle cell, there's a plasma membrane of the muscle cell, and from those plasma membrane are these sort of invagination called T-tubules. They're sort of a continuation of the plasma membrane. Those T-tubules come in close contact to the sarcoplasmic reticulum. Right? Inside the sarcoplasmic reticulum is calcium. Your muscle cell is also innervated. So there's a nerve touching the plasma membrane. So during muscle contraction, there's a nerve impulse. There's an electrical charge coming down the nerve, and that electrical charge is then transmitted to the plasma membrane of the muscle cell, and that electrical charge keeps going down the plasma membrane, down the T-tubule. So the T-tubule is carrying the electrical charge from that from your nerve that you've triggered to move your muscle cell, and that T-tubule is then going to come in contact with the calcium release channel that's part of the sarcoplasmic reticulum. So your nerve impulse is going to open this calcium release channel, and there's high levels of calcium inside the sarcoplasmic reticulum, very low levels of calcium in the cytoplasm. So calcium is going to come out of the sarcoplasmic reticulum, come into the cytoplasm. It's going to play a role in muscle contraction. We'll see how in the next slide. As soon as the calcium comes out, it's triggering a muscle contraction, but almost instantly it begins to get pumped back into the sarcoplasmic reticulum by a calcium ATPase. So we'll talk later about this calcium release channel and this calcium pump. We'll talk about the membrane transport later on. But for now, a muscle impulse releases calcium from the SR. It's going to come out, trigger contraction, and then very quickly get pumped back into the SR, into the sarcoplasmic reticulum. Calcium is very abundant outside of the cell. There's lots of calcium outside of the cell. That's why you make bone outside the cell. There's very little calcium in the cytoplasm. Calcium can be used as a secondary messenger. Calcium is used in cytoplasm for sort of small jobs, but you don't have a lot of calcium in the cytoplasm because you have a lot of phosphates in the cytoplasm. You have ATP, GTP, etc. Calcium and phosphate tends to make a mineral, which is what bone is. So you don't want any sort of calcified stuff inside the cytoplasm. So when you have calcium in the cytoplasm, where you have all these phosphates, it's only there for a short time. So in this case, calcium is released, it does its job in muscle contraction, and it's quickly pumped back into the SR. Make sense? Okay, so your nerve impulse travels to the plasma membrane of the muscle cell, down the T-tubule, releases calcium. That calcium now is going to be able to flow all over the sarcomere, and it's going to interact with these proteins called troponin and tropomodulin. So the actin filaments are not only coated by a number of proteins, they're capped by tropomodulin and CAPZ, there's nebulin, which we just talked about, but they also coated by this protein called tropomyosin and troponin. Okay, there are different forms here. You can see here there's troponin T, I, and C. You don't have to worry about the variants. Troponin is a calcium binding protein. Okay, so troponin will bind calcium. And when troponin binds to calcium, it shifts tropomyosin. So tropomyosin and troponin are coating the actin. In the absence of calcium, when there is no calcium, troponin and tropomyosin are covering the myosin binding sites. So the myosin fixed filament, the heads, normally bind to actin, but they bind in specific locations. Those locations are covered in the absence of calcium. So when there is no calcium, when you don't want the muscle to contract, the myosin head binding sites are covered by troponin and tropomyosin. When calcium is released, calcium binds to troponin. Troponin causes a conformational shape of tropomyosin. The whole structure of troponin and tropomyosin moves along the actin microfilament, and it exposes the myosin binding site. So now myosin can bind, the myosin head can bind. Once the myosin head binds, it can then do a pyrostroke, and you can have myosin movement. All right, so during striated muscle contractions, there's two cycles going on that somehow in your head you got to get them straight. One is ATP and myosin moving along, along actin, right? So myosin in the state of rigor, in the rigor state, has ATP bound to it. So when myosin is in rigor, myosin is bound to ATP in rigor. When ATP binds to it, myosin lets it go. In that time that myosin lets it go, potentially troponin and tropomyosin can cover that binding site, and now it has to wait. Okay, so there's two cycles, one of myosin letting go and binding to actin as it takes the step, and the other cycle of calcium coming in and out, covering and uncovering the two, the myosin binding sites. So there's two cycles going on here, the ATP hydrolysis cycle with myosin stepping, and calcium being released and quickly pumped back in to stop the contraction. Okay, so you have to try to think about what state is the myosin head in when calcium is being released. The myosin head wants to bind, right? So the myosin head is going to be in the ATP-PI state, and then as soon as the binding site is released, it can bind, it can undergo the power stroke, then it lets go. Once it lets go, potentially then if calcium is pumped back, the contraction stops. Right? So that's striated muscle regulation. Hopefully it makes sense. You can think about the two cycles, the ATP hydrolysis and the calcium release, and hopefully they all make sense to you as well. Okay, so that's actin functioning in striated muscle. Now we're going to look at actin and myosin functioning sort of outside of striated muscle. So we're going to talk about several examples of actin and myosin in non-striated, non-skeletal muscle. Okay, so we're going to mention, I think, 5 examples of function of actin and myosin in non-skeletal muscle. Right, so the first one we're going to talk about is cytokinesis. We already sort of mentioned this occurring. We know that during metaphase, you have the polar microtubules. Those polar microtubules are going to localize Rho protein. Rho protein is going to be localized at the metaphase plate. We already know that Rho is involved in actin polymerization. So this contractile ring that's present at the metaphase plate is an actin and myosin structure that actually, when you look at it, looks like a little sarcomere. It has repeating units. They look like sarcomeres and they function like sarcomeres as well. These sort of structures are found in other places that we'll talk about. So this contractile ring that's found during cytokinesis is made up of actin and myosin that sort of forms this sarcomere structure, and it's in a ring. Later on, we'll talk about these things called adherence belts, which are found at the cell surface of cells. Those are also contractile, so you can contract these rings, and you can change the shape of a cell by contracting this ring. There's another structure called a stress fiber. Stress fibers involved in cell migration, which we might talk about today. They are also contractile. So there are other places where we have these simple sarcomeric type of arrangements that are involved in contraction. In the case of the contractile ring, that contractile ring is getting smaller and smaller and smaller as cytokinesis is going on. So unlike striated muscle, where this sarcomere is quite stable, here the structure is constantly being depolymerized. So as it's contracting, it's becoming smaller as actin is being depolymerized and myosin is being removed. So while they look similar, these are transient structures that are sort of present for a little while and that can be removed as contractions occur. All right, so actin and myosin also form these sarcomere-like structures and other cell types in addition to striated muscle. An example here is the contractile ring during cytokinesis. Pretty simple, hopefully. All right, actin and myosin also function in smooth muscle. So smooth muscle contraction is different than striated muscle contraction in a number of ways. One is that it's not voluntary. It's not controlled by nerve impulses, and it says down here. It's actually regulated in a number of different ways. You have different types of smooth muscle in your gut and other places, and they're regulated in slightly different ways. But the regulation is not through a nerve impulse. The other difference is that it doesn't involve troponin and tropomyosin, so it doesn't involve uncovering a binding site. So the contractions that are provided by smooth muscle are more persistent than skeletal muscle. With skeletal muscle, if you want to keep on contracting your muscle, you're going to have to keep on releasing calcium and keep on sort of contracting, whereas here the mechanism is different, so the contraction is much more stable, because here contraction is triggered by activating myosin. Okay, so smooth muscle does not have troponin, so it doesn't have that calcium binding protein called troponin. Instead, it has a different calcium binding protein called calmodulin. Okay, so calmodulin is going to respond to calcium signals. So this calcium can come from the sarcoplasmic reticulum or from the ER in the smooth muscle. It can come from external sources. There's different places where this calcium can come from. But what this calcium does is it actually phosphorylates the regulatory light chain. And it's the phosphorylation of the regulatory light chain of myosin that activates it. So now you have an active myosin that can act with actin to provide contraction. Okay, so there's a number of different actin and myosin structures that are found in smooth muscle, but they are activated because calmodulin is phosphorylating this regulatory light chain, and this light chain then activates myosin, and now this myosin stays active so it can keep on contracting for long periods of time. Okay, so this isn't voluntary, it's regulated by a number of other mechanisms. So smooth muscle contraction is different than striated muscle. It's not voluntary, it doesn't involve troponin, it involves calmodulin, a different calcium binding protein. And you're activating a light chain, you're phosphorylating the light chain, and once active, that myosin stays active for a long time, and the contraction can be persistent. Make sense? All right, so cytokinesis, smooth muscle contraction. Another job for actin and myosin is vesicular transport. We mentioned myosin 5. Myosin 5 was the myosin that had the largest step size. It is a motor protein that can move almost anything. So the cargo that this protein can move is quite varied. And it's best seen as an example in budding yeasts. So yeasts are eukaryotic cells that can undergo mitosis in a number of ways. There's fission yeast and budding yeast. And in the budding yeast, you grow this sort of bud. Remember, yeast have a cell wall. So you grow a little bud, and then into this bud, you have to transport everything so that this new cell, when it undergoes mitosis, will have all the components you need. So you have to put into it all the components that's found in the mother cell. So you have to put in everything from mitochondria to peroxisomes to vacuoles to ER to everything. So, and this is all done by myosin 5. It's also a nice example of how during budding yeast mitosis, the actin network is set up to allow transport into this new bud. Right? So the way it works is you form a bud, and in that bud you then polymerize actin such that all the actin plus ends are leading to the bud and then into the bud. And this is done by using formin and Rho-GTP. So there's a little movie here that hopefully will show that. So there's going to be a lot of labels here, a lot of proteins that stabilize, a lot of proteins that are going to do different things. The names are going to be slightly different. Sort of ignore most of it, just try to follow along with what I'm going to say. So the idea is you're going to try to transport stuff to the bud and then into the bud. So you're going to put the plus end of some microfilaments at the entrance of the bud and the plus end of other microfilaments deep inside the bud. So you'll see that you'll have rho GTP and formin present here. This will be the word plus end is. And rho GTP and formin present here. And then from that rho GTP and formin, you're going to polymerize actin. So let's hope this works. Right, so there's all kinds of proteins. That's the formin, that's rho GTP. So you have formin and rho GTP present, leading to the bud and in the bud. And then you're going to polymerize actin there. So actin is going to polymerize, and that's where the plus end of the actin will be. And actin, as it polymerizes, the minus end is going to grow away because you're adding to the plus end. So actin is going to grow away. It's going to be stabilized, but never mind all of that. So you have the plus end leading to the bud and in the bud, so the minus end is growing away. So then once you have the minus end that goes deep into the cytoplasm, you can use myosin 5 to move cargo from the minus end to the plus end, and then transfer it into the new minus end of the microfilament inside the bud and go towards the plus end. So you're always moving towards the plus end to get to the bud and to get into the bud. So there's myosin 5 taking some cargo in. It invites its friends. And you're transporting everything. So you can transport cargo, but you can transport everything, including, as I said there, peroxisomes and RNA. Eventually, you have to place the nucleus in a position such that when mitosis occurs, half the DNA goes into the new bud. Importantly, though, here you have myosin 5 walking on actin and also binding to microtubules. So microtubules are pulling on the nucleus. So here you have a microtubule as cargo, and you have to kind of imagine that anything can be cargo, including other cytosolic proteins. So microtubules can be cargo for actin, actin can be cargo for microtubules, right? Another important thing to mention is that, like a lot of motor proteins, myosin 5 is not active unless it's down to the cargo. There's no point in the motor protein moving to the plus end unless it's down to cargo. So it's only going to be active when it's down to cargo. Okay, so myosin 5 is an important cargo moving mechanism inside cells. It can move almost anything, and a good example of that is looking at yeast budding, where indeed it does move everything literally into the new budding yeast daughter cell. Okay, another example of actin and myosin that's more important in plant cells than in us is something called cytoplasmic streaming. So as cells get bigger, diffusion becomes a problem. So for a lot of our cells, waste gases and sort of things can diffuse in and out because most of our cells are quite small. As you get into larger cells, which a lot of plants have quite large cells, so diffusion becomes a problem. There's too much space for things to diffuse in and out quickly. So in lots of plant cells, you have something occurring called cytoplasmic streaming. Cytoplasmic streaming is the fact that the cytoplasm is being moved around on purpose. So if you can imagine if cargo is being transported, so whether kinesin is moving something or actin is moving something, or myosin I should say on actin, when you move things through the cytoplasm is going to move. So plant cells do this on purpose. We already know that actin is cortical, actin is around the outside of the cell, and indeed plant cells have this cortical actin that surrounds the entire cortex of the cell. It goes all the way around. And you have motor proteins on this cortical actin, and these motor proteins are constantly moving, and they're constantly moving the material inside the cytoplasm, so they're bound to the ER and everything. So the cytoplasm of plant cells is constantly moving around to help with diffusion. So waste gases can get out faster, good gases can get in faster. All right, there's a bit of a movie here that shows that, just to kind of show you what it looks like. Hopefully it'll work. There is narration, but it's not working. Well, regardless, you can see that
the cortical cytoplasm containing the chloroplasts is stationary, while the plasma streaming over it sets the cell sap, including its incorporated bodies, in rotary motion. Cytoplasmic streaming of this variety is promoted by actin filaments, such as here in the tip of a root hair of linoleum.
So different plant cells do it in different ways, but basically the cytoplasm is constantly moving because myosin and actin are constantly moving organelles around. Okay? And then the last example, which is going to take a while, of actin and myosin in non-muscle cell is cell migration. Right? So cell migration is common during embryogenesis. So when you're very little, you get all kinds of cell migration going on. There's processes called gastrulation and neurulation, solidogenesis, neural crest cell migration. Lots of things move around as in an embryo. In an adult, there's very little cell migration that occurs. Your immune cells can move around a little bit. During pregnancy, there's some remodeling that occurs where cells move around. If you injure yourselves, there's a lot of repair cells move around. But in general, there's little cell migration in adults. When cell migration does occur in adults, often it's bad, such as during cancer metastasis. So trying to understand cell migration in embryos kind of helps us understand what goes wrong later on where things are moving in adult bodies where they shouldn't be. Right? So for a cell to migrate, you have to imagine it needs a substrate. It needs to bind to something. It needs traction to move. So migrating cells, when you look at the actin network, they have sort of specific structures that are recognizable because it's using actin and myosin to move. So the migrating cell is going to push membranes forward. So as a cell is migrating, it's pushing membrane forward. So there's membrane extension, and you make these things called lamellipodia and filipodia, which we'll talk about in a second. So there's membrane protrusions because actin is being polymerized. So actin is being polymerized at the front end, it's pushing the membrane forward, you get the lamellipodia and filipodia, and then they look to attach. Okay, we'll talk about that. But when they form an attachment, that attachment is called a focal adhesion, and we'll talk lots about focal adhesions later on. Focal adhesions use proteins called integrins. Integrins bind to proteins outside of cells. So all multicellular organisms have extracellular matrix, ECM. There's many different types of proteins out there. A lot of these proteins can be used for cell migration. So all of our cells are surrounded by ECM. Everyone's heard of collagen. People may have heard of fibronectin, there's laminin, thrombospondin. There's lots of proteins out there that can be used for migration. So these proteins called integrins are going to bind to this extracellular matrix and provide that traction. And when the integrins bind to this extracellular matrix, that's called a focal adhesion. Make sense? So the focal adhesion is like little sticky feet that the cell has. So on the outside, the integrins are bound to the extracellular matrix. On the inside the cytoplasm, the integrins are bound to these actin and myosin structures called stress fibers. So there are stress fibers inside cells that connect focal adhesions, and those stress fibers are basically little sarcomeres. They're arranged of actin and myosin, and they're contractile. So a migrating cell is doing two things with the actin cytoskeleton. One is it's polymerizing it at the front end, pushing the membrane forward, and then it's forming little feet. Those little feet are focal adhesions. And those focal adhesions are connected between them by these structures called stress fibers, and those stress fibers are actually contractile bundles of actin and myosin. So for each stress fiber, at both sides are going to be a focal adhesion. So there's a focal adhesion at the front side, or at the back side here, a focal adhesion at the front side. And those focal adhesions connect stress fibers, and those stress fibers are contractile. And we'll see how that works in a second. Right? So for cells to migrate, they need a direction. They need some sort of polarity. They need to know which way to go, and that often involves chemotaxis. So chemotaxis is a response to some sort of chemoattractant. It's going to go in a certain direction because there's some sort of chemical that it wants. right? So in development, there's all kinds of developmental cues. With your white blood cells, there's all kinds of immune cues that tell the leukocytes or whatever come in this direction to fight the bacteria. So there's going to be some sort of chemotaxis to tell the cell which direction to go to. And because of that, the cell will have a polarity. The cell will have a front end and the cell will have a back end. Okay, so there's a lot of little things here. What's the important thing to appreciate with what's going on here is that if a cell is going to respond to a chemoattractant, so if you have an immune cell, you have a white blood cell sitting there in your tissues, and it's waiting to go off in a different direction to fight an infection, it has to be able to respond to go in any direction. So if you have a bacteria sitting in one side, and that bacteria is going to trigger an immune response, and it's going to release some sort of cytokines that are going to go to the cell, that immune cell has to have the receptor along its entire surface. So the receptor for receiving this chemoattractant has to be along the entire surface of the cell so that the cell can respond in any direction. So if this was an immune cell, there's a pathogen on this side, it would know to go this way. If the pathogen's up top, it knows to go this way. So the receptor has to be present on the entire surface so that a signal from any side can be received. That signal is going to trigger actin. The first thing it's going to do is it's going to trigger actin polymerization. Actin is going to polymerize and push the membrane in that direction. So not only does the receptor have to be present on the entire surface, but the appropriate G proteins have to be present along that entire surface. So we've already talked about Rho and RAC and CDC42. They have to be present everywhere if the cell can move in any direction. So the receptor will be activated in a certain location. It's going to activate G proteins that can then cause actin polymerization in any direction. Okay, you can largely ignore a lot of this other stuff, but just to show you an example movie, I hope. So this is a cell, and cyclic AMP is being added. Cyclic AMP is a chemoattractant for lots of cells. Cells love cyclic AMP. And you'll see the cell is going to respond to cyclic AMP, and because it has a receptor on its entire surface, it's going to go in that direction by polymerizing its membrane in that direction. And then as you move the signal, you can see that the cell is going to respond. So it's going to go towards the little pipette tip, and then just mess with it for a while. At some point, you've dumped so much cyclic AMP in there, there's no more gradient of chemoattractant, but you can see that the cell can respond in any direction because it has receptor for cyclic AMP in this example on the entire membrane, and it can polymerize the actin cytoskeleton in any direction. With most cell migration, always consider that the cell has to move on something. It needs traction, right? So the cell has to be moving on something. Okay, so let's kind of look at cell movement a little more specifically. Okay, so we're going to look at some steps of cell movement. We're going to look at a video, and then we're going to look at the steps of cell movement again, and hopefully you get it. All right, so what is this showing you? This is showing you some sort of substrate. So this cell is sitting on some sort of ECM substrate here, and it's sitting on these things called focal adhesions. Okay, so if you remember, focal adhesions are these protein called integrins. The integrins are binding to the extracellular matrix on the outside, and they're binding to these things called stress fibers on the inside. So, right? So this is sort of looking at the cell from the side. If you look at the cell from the top, it has these stress fibers that are sort of linking these focal adhesions. These diagrams are a little bit different, but let's just kind of draw some stress fibers inside this cell. Okay, so different cells, we're going to form focal adhesions in different ways. In this example here, these focal adhesions are forming these long stress fibers, and the movie's running wild. Never mind the movie for now. So this cell has long stress fiber. This cell has stress fibers that link all these focal adhesions. Okay, so focal adhesions are places where there's feet, for lack of a better word. And in between all of those focal adhesions are stress fibers. So the cell is sitting on focal adhesions. In between those focal adhesions are stress fibers. So in this example, there's 1, 2, 3, 4, 5 focal adhesions. And there's 1, 2, 3, 4 stress fibers in between them. In this example down here, there's only focal adhesions at the back and at the front, and there's long stress fibers. So this cell is basically sort of one long stress fiber. This one, there's 4 of them. So different cells can do things differently. Okay, so the cell is sitting there, it's going to get a chemoattractant. Something is going to trigger it to move in one direction. There's going to be a chemoattractant. The first thing it's going to do is going to polymerize its actin cytoskeleton in that direction, and it's going to push the membrane forward, and it's going to push forward a filopodia, which is a thin membrane protrusion, or a lamellopodia, which is a thick membrane protrusion. So it's going to push the membrane. It doesn't really matter if it's a little membrane or a large membrane, it's the same thing. That membrane is going to search for a new place to stick. When that membrane forms a new place to stick, it's going to form a new focal adhesion. When it forms a new focal adhesion, it's then going to form a new stress fiber between the old focal adhesion that used to be the front of the cell, and now there's another one. So now you have a cell that has 6 focal adhesions and 5 stress fibers. You've extended forward and you've made another focal adhesion and another stress fiber. So after this, the cell hasn't really moved yet. So far it's just kind of extended and stuck to a new location. Now it's going to translocate its body. It's going to move the body of the cell forward. Okay, so there's lots of actin and myosin contraction. These stress fibers are contractile. These stress fibers do contract and play a role in this, but there's other actin and myosin interactions that are moving the cell body forward. So once you move the cell body forward, you basically let go of the old focal adhesion. You release the old focal adhesion. Make sense? I hope. So let's just try this movie here. Okay, so this movie, there is audio, but I don't know why it's not here. There are similar movies in Achieve. Where the heck am I with this thing? Oh. Okay, so it's suggesting there's actin in one part, myosin in the other part. There's actin and myosin in most parts of it. The first thing that's going to happen is you're going to have actin polymerization, and you're going to push the membrane forward because of actin polymerization. So the membrane is going to be pushed forward, actin is going to polymerize. So actin can polymerize using formin to form single filaments, or it can use ARP23 to form branched filaments. So as it's pushing the membrane forward, it's going to form a new focal adhesion. We have old adhesion sites, and it's going to form a new one here. Once it forms this new one, you're going to get another stress fiber. So there's going to be a stress fiber going from here to here, here to here, here to here. Then there's going to be a contraction event. I don't know what this movie's trying to show. There's myosin here, and somehow myosin is doing this. I don't know what that is. It doesn't really matter how myosin and actin is doing. There's a lot of studies that still can't quite explain how the cell body is translocated forward. There's a lot of contractile structures here, but focal adhesions are fundamental to this. Focal adhesions are how the cell sticks, and then they are contractile, and they help things move forward. Make sense? Okay, so looking at that, then you think, all right, how many flat surfaces are there in your body? 2-dimensional flat surfaces in your body. Answer is 0. There is no flat 2-dimensional surface in your body. Your body's 2-dimensional, so you have to think of this in 3 dimensions, right? So it's not flat, it's sitting and surrounded by stuff. So you form focal adhesions all around the cell. You have a cell and there's focal adhesions top, bottom, and other places. And then things make more sense where you protrude forward and you have focal adhesions going all the way back, and you can almost think of it like a tube of toothpaste. You kind of squish things and you're squishing things forward to some extent. So focal adhesions don't quite work like this. cells don't like being on flat surfaces. That's why they kind of drawn this cell sort of with the cell kind of lifting itself up. They do that because when you culture cells on a flat surface, they don't like it. They're trying to get themselves off this stuff. So focal adhesions are used. It just doesn't quite look like this, but the concept is the same, that you're protruding the membrane using polymerization, you're forming a new focal adhesion, that new focal adhesion forms a new stress fiber, and then those stress fibers contractile and allow movement. Make sense? Easy, I hope. All right, we talked about this is all regulated because you need the receptor on the entire surface of the cell, and then you need to activate polymerization. You need to have these small GTPases, these Rho proteins present everywhere. Okay, so you have the plasma membrane of the cell, and you're going to be responding to some sort of chemoattractant. So different cells are going to be attracted by different things, but they have to have that receptor present on all parts of the cell surface in order for that cell to move in that direction. Hopefully that makes sense. Okay, so you're getting a ligand binding to the receptor, then that receptor has to activate actin polymerization. So you need these proteins that we've already talked about, CDC42, RAC, and Rho. These are Rho proteins they're all GTPases. You know they have to be in the GTP form to be active. So in the GTP form, they can activate formin or they can activate ARP23. So in response to the chemoattractant binding to the receptor, it's going to activate these proteins. And once these proteins are activated, they're going to trigger actin polymerization, either through formin or through ARP23. So far, so good? What do they do? Well, what you can do is you can make dominant active forms of these proteins. So normally they're only active in the GTP form, but you can make a mutation that's always active to see what does this protein do when it's always active. So here you have a cell, and it's fluorescently stained for actin. So wherever you see sort of this white glow, this white color, that's what actin is. So in a controlled cell, when none of these proteins are activated, these cells aren't really migratory. Actin is cortical, so most of the actin is present at the plasma membrane, sort of close to the outside. There's other actin in the cytoplasm, but most of the actin is cortical next to the plasma membrane in a controlled, unmigrating cell. Then you introduce one of these proteins in a dominant active form. So dominant active is always on, right? So you activate Rho, so the dominant active Rho. Rho, if you remember, can activate formin. When you activate Rho, always, you get what looks like stress fibers. So stress fibers are regulated, looks like it, by Rho activation. You can do the same thing with dominant active RAC. So both RAC and CDC42 activate ARP23. If you activate RAC, dominant active RAC, you see that there's membrane ruffling all around the surface. So all of the membranes are pushing forwards. You're basically getting networking all around the cell surface. There's ruffling and protrusions everywhere. So you get networking occurring up in every direction all at once. So RAC looks like it's forming these large networks all around the cell. When you use dominant active CDC42, you get these little small protrusions that look like filopodia. So with RAC, looks like you're just making lamellipodia everywhere. With CDC42, these are small protrusions, look like you're making filopodia, small membrane protrusions everywhere. So you have CDC42 making filopodia, RAC making lamellipodia, Rho making stress fibers, which are basically all the components that are involved in cell migration. That good? And you need all three of them working together. So you need the three of them to kind of have the most robust cell migration. And you can see that by doing something called a scratch closure wound assay. So you can take cells and you grow them to confluence. So these cells are now covering the dish that you've plated them in, and they can't move anywhere. then what you can do is you create a scratch. So you clear out a little area and you allow them to close this wound. So they're going to migrate to close this wound, and you can measure how fast these cells move. So here's an example of what it looks like. You make a little scratch, and then slowly over time the cells move into that scratch to close it. So if you do this experiment and you see how much of that scratch is closed, if you have control cells, so cells that have all of these proteins, so cells that have CDC42, RAC, and Rho, they basically can control, they can close 100% of the wound. So cells that have these three proteins migrate and close the wound, no problem. However, if you mutate one of these, so now these are dominant negative, these are the proteins that don't work. So if you mutate RAC or CDC42 or Rho, you get very little wound closure. These cells have trouble migrating. So you need the three of them working together to get sort of the most robust, the best cell migration. Okay, so these three indeed work together, and there's signals between them. So there's a lot of things going on here. We're just going to concentrate on sort of the simplest things, for lack of a better way to put it. Okay, things tend to start with CDC42. So there is receptor present for some sort of chemoattractant cytokine that's going to come in. It's going to bind to the receptor, and it's going to activate CDC42. So CDC42 is typically considered the first thing that's activated. You can see that RAC is also activated, but CDC42 is sort of the prime one. CDC42 is going to turn on a gene called PAR6, which we'll talk about on the next slide, and this is going to be involved in cell polarity. So CDC42 is establishing cell polarity. It's saying this is going to be the front of the cell. We're moving in this direction. CDC42 is also going to work with WASP to activate ARP23, something we already know it can do. And you're going to get actin polymerization, and you're going to form filopodia. Okay, so this is using ARP23 to form filopodia. You might think filopodia are sort of bundles, but the filopodia can be made with ARP23, and indeed that's what's happening here. Okay, so CDC42 is the first thing activated. It's going to set up polarity. This is the front end, and it's going to activate WASP and ARP23 to form filopodia. And you're going to get these little membrane extensions. At almost the same time, RAC is going to be activated, and it's going to work through wave to also activate ARP23. And here you're going to get lamellipodia formation. You're going to get large chunks of membrane moving forward. So different cells use different amounts of filopodia and lamellipodia to migrate. But often these two pathways work together. Okay, so you have membrane protrusion. Once the membrane is protruding and then the cell starts finding new places to stick, you then get activation of Rho-GTP. Rho-GTP is going to do two things. It's going to activate formin, and formin is going to cause actin polymerization to start forming those stress fibers. But also, Rho is going to activate myosin. So myosin is going to get in with those stress fibers, and those stress fibers can become contractile. Right? So the lamellipodia and filopodia are not contractile structures, but the stress fibers are, because Rho can activate both formin to cause actin polymerization. It can also activate myosin, so that myosin is a functioning motor protein. Make sense? Okay, so you have things being migrated, usually at the front end. CDC42 is the first one. It's going to activate the front end. It's going to activate this gene called PAR6 that gives it polarity. So this is the front end of the cell. Next, you're going to activate RAC. RAC is going to be involved in making lamellipodia. And then eventually you're going to activate Rho, and then Rho is going to activate myosin, and you get the stress fibers, and you get the cell movement. So it's usually the order is CDC42, RAC, and Rho. Okay? You have the front end polarity. There's a lot of stuff here. The only thing I want you to take from this part is the front end of the cell is polymerizing actin. The front end of the cell needs actin monomers to push forward. you need to get actin to the front part of the cell. So one of the things this gene, this PAR6 is doing, so CDC42 and PAR6 is polymerizing microtubules. So microtubules are polymerizing to this front end so that the microtubules can transport actin to that front end. So the microtubules and kinesin are going to transport actin monomers. There's not a whole bunch of other stuff because you need tons of actin to this new growing front end. So you have to also involve the microtubule cytoskeleton. So CDC42 is not only causing actin polymerization, but it's also through PAR6 causing microtubule polymerization to get stuff there. Hopefully that makes sense. See you guys Wednesday. Okay. Tell me what we attached to it.
Yeah, I don't know how to attach it.
What? Extracellular matrix. Collagen, fibronectin, laminin. Proteins in it. The cells secrete those proteins. So around all of your cells are extracellular matrix. Collagen, fibronectin, laminin. So that's what they move on. They move on proteins that are found outside cells. Look at the grades. Yeah, just keep these things.
Dude, I don't know.